Now here you see this part was left in the yesterday's class. Now we will be dealing with ribosomes. Now basically what are ribosomes? Ribosomes, they are the site for protein synthesis. Okay, the protein synthesis, ribosome plays an essential role. Without ribosome, protein synthesis cannot happen. So if protein synthesis does not happen, the total genetic reaction will not be able to happen only because of the absence of protein synthesis. So basically ribosome is a very vital organ, and this is not a membrane bound organ, and it is present both in the eukaryotes and prokaryotes. Now they are associated with the plasma membrane. That means they remain attached or nearby the plasma membrane. Now two subunits are there in the ribosome. One is the 50S and another one is the 30S, but together they form a 70S ribosomal unit. We call it as a bigger unit, and this is a smaller unit, and together it is a 70S unit. And ribosome is always attached to mRNA strands. Okay, so basically when they are arranged to mRNA strand, they form a polyribosome or a polysome. Okay, polysome and polyribosomes. And other things that are present as cell inclusion or inclusion bodies, these basically they are not bound to any membrane, but they lie free in the cytoplasm. Okay, like granules. What are they? Phosphate granules, then glycogen, etc. These are all known as cell inclusion. Now here we again come to the very important part of the chapter, that is known as eukaryotic cells. Now eukaryotic cells is basically all the above organism above the monoreans, algae and monoreans. Some of the algae are prokaryotic, but rest of them are all eukaryotic cells. That means they are membrane bound. Okay, so what is the basic principle attached to the eukaryotic cell? They show compartmentalization. What is compartmentalization? Every organelle that is present within the cell have got a very discrete section. Okay, discrete sections or compartments, separate unit, and they do not mix one with the other. Now see, they are all membrane bound structures. This is also a very important difference between the prokaryotic cell. So here the eukaryotes, they all are membrane bound structure. Then they have got a complex locomotory and cytoskeleton structure. Earlier we have seen that the eukaryote, in the prokaryote, they had got basal body, cilia, flagella, fimbriae, okay, and pillae. These were the locomotory organs of the prokaryotes, but here they have got a complex locomotory organ, and that or the cytoskeleton structure. Now next is the genetic material. Here we do not find genetic material just scattered in the cytoplasm. These genetic material, they are organized into a chromosome, and these chromosomes are neatly packed and present within a membrane bound nucleus. Okay, so distinct nucleus with a chromosome, genetic material, and basically genetic material here in all the eukaryotes are DNA. RNA is not a genetic material, but they are used as, we have mRNA, messenger RNA, and transcripting RNA, tRNA and mRNA. Okay, and what is the and now these eukaryotic cells are distinctly divided into plant cell and animal cell. These are also distinctly differentiated. Now here I'll give you a notes of plants and animal cells later on, but here roughly the plant cell and animal cell, you see it's a very common differences, and you have done in your earlier classes also. See, cell membrane is present to both the plant cell and the animal cell. Cell wall is present only in the plant cell, whereas cell wall is absent in the animal cell. The vacuole, the plastids, these are present only in the plant cell and they are absent in the animal cell. Then endoplasmic reticulum, mitochondria, Golgi body, these are all common to both plant cell and animal cell. But here you see centriole and centrosome. Centriole, centrosome and lysosome, these three are prominently present in the animal cell, which is entirely absent in the plant cell. Okay, so this is some of the basic differences between the plant cell and the. Now the next topic what we will be dealing is the cell membrane. Okay, now basically cell membrane is something that is both present in the prokaryotes and eukaryotic cell, but a well-defined membrane basically consists of the lipoprotein. Now what is lipoprotein? They are made up of lipid and protein. Okay, so this presence and the combination of lipoprotein makes it selectively permeable. Selectively permeable, that means what? It allows certain substances to pass through it and does not allow certain substances to pass through it. Now lipids, basically if you see a lipid molecule, lipids are made up of a polar head and a non-polar tail. Okay, now you see the structure, the round globular portion, this is known as the polar head, and here this is the non-polar tail. Now polar head is characteristics by hydrophilic in nature. What is hydrophilic? Hydrophilic means water loving or any other fluid loving. And non-polar tail, it is hydrophobic. Hydrophobic means what? It is water hating. That means it does not repellent rather. Okay, hydrophobic is water repellent and it does not want the structure to, you see, it does not allow the liquid to pass through it. Now here this is the structure. Now you see this red, this is the bipolar head. This is also, see, it is a lipid protein bilayer, 2 layers of lipid molecule, and here you see this is, it is an intrinsic protein layer that is in a big molecule here. Now you see this head, these are all hydrophilic, that means attracting liquid. So liquid comes here. And see the tail portion, they are hydrophilic, that means hydrophobic, sorry, that means they are water repellent. So it does not come from here, that means it cannot enter the cell, whereas it passes through a small gap here between the tails. And this integrated protein structure, you know, it rotates in a circular manner. For example, it wants to transport some molecule, for example, a larger glucose molecule within the cell. So if, see, this is the molecule, if the sugar molecule sticks here, this will suddenly come down and this portion will go up. That means it would rotate. So that means it would allow larger molecules to get in. Okay, so this is also a part by which it can transport, and sometimes it is an active transport. That means energy used to transport against the potential gradient. Now to another method by which the membrane absorbs outside material is through osmosis and diffusion. These are physical process of transport. Osmosis is when two different fluids, they are separated by a semi-permeable membrane. Molecules from the region of higher concentration moves to the region of lower concentration, and again simply diffusion is molecules from the region of higher concentration moves to the areas where the concentration is low. So these are the two physical processes, and here you see active transport means energy used to transport against the potential gradient, and passive transport, that means can be transported by using the natural gradient. For example, osmosis and diffusion, they are all passive transport. Now if you can see in this figure, you can see this is a fluid mosaic model of the plasma membrane. See, the integral protein molecule can be seen. Then like beads like structure, you can clearly see the protein molecules with the, that is known as the hydrophobic and hydrophobic tail and hydrophilic head, and you can see the gap between these two from which the fluid actually passes through. This area you can see here. Okay, now this is basically, see, this is the fluid mosaic model as I told you, and it was founded out or invented by Singer and Nicholson. Okay, the fluid nature of the membrane is also important from the point of view of the function like cell growth, formation of intercellular junctions, secretion, endocytosis. Endocytosis is pinching in of substances, and cell division, etc. Active, passive, osmosis, these all I have told you. Now here you see, these are the two basic diagrams of a plant cell and animal cell. Majority organs you see, vacuoles are present here, vacuoles are not present here, centrioles are present here, centrioles are not present here, ribosome is present in both. The cell wall is here, but here they don't have a cell wall. Okay, so you have to go through it and try to draw the diagram